Good morning, good afternoon, everyone. Hi, I am Rosario Labruto with Sandoz Pharmaceuticals, and today I will be presenting on advancing development of complex generics to improve patient access to medicines. I would like to thank the FDA for the kind invitation to present in this exciting kickoff session, the next 5 years of the Generic Product Science and Research Program. Complex generics represent an untapped savings opportunity for the U.S. healthcare system, including patients, Medicare, Medicaid, and commercial payers. Despite recent efforts to promote the approval of complex GX, these products are still slow to be approved, and more needs to be done to advance and enhance access of these critical medicines for patients. Complex products are medical products where complexity or uncertainty concerning the approval, pathway, or possible alternative approaches would benefit from early scientific engagement, such as products with complex active ingredients and drug-device combination products. These products are, in general, harder to develop with traditional bioequivalence methods, and therefore fewer exist, resulting in less market competition for these products. This is where we, as an industry, can get more involved and potentially steer the conversation to better informs FDA thinking around product development and innovation for complex products. In the next 5 years, a combined total of approximately $90 billion in U.S. pharma sales are at risk from patent expiration, with approximately $14 billion stemming from the injectable segment. Injectables represent approximately 25% of the total number of LOE opportunities compared to ophtha and nasal, for example, each at less than 5%. If we look at the injectable segment, there are multiple complex type of generics such as long-acting release, like suspensions and microspheres, liposomal products, suspensions, iron colloids, polypeptides, and oligonucleotides, with a large part of the LOE value being derived from peptides and oligonucleotides. To put into perspective, there have been approximately 13 approved oligonucleotides from 1998 to 2021, mainly in the orphan space, with exception of inclisiran. 80 compounds are actually currently in phase 2 and 3, and 130 clinical trials are ongoing. And for peptides, greater than 80 were approved, with 8% of FDA branded drug approvals being for peptides during 2016 to 2021, and currently 170 clinical trials with peptides are ongoing. Each of these subgroups has their own complexities, and we will go over some of the challenges and opportunities for advancing generics in these categories, and also my colleague, Manashki Jain, will dig a bit deeper into the oligonucleotides and ophthalmic products in session 6. Generally, more complex dose delivery forms have on average fewer competitors per product with higher barrier to entry due to advanced technical capability requirements. The development of complex GX products has multiple challenges stemming from material complexity such as API and excipients, formulation complexity, manufacturing process complexity, analytical methods, and in vitro BE complexity. For example, where multiple orthogonal methods would be needed for comparability studies and to demonstrate sameness, and challenges with sample preparation due to formulation matrix interferences. Interpretation of data in comparison to RLD, and access and availability to the desired CROs, equipment, and resources skilled in the art of developing complex generics. Regulatory complexity and biocomplexity, for example, demonstrating bioequivalence due to high inter or intra-subject variability when such studies are needed. And lastly, access to RLDs. The big question here is, could utilizing ex-US RLDs be advantageous in further advancing the development of GX products for the US market? Now, let us take a look at trends toward complex GX. So one of the first trends here is where delivering more value to patients by addressing unmet needs and enabling market differentiation. As market becomes more saturated with standard generic products, some companies are moving focus to complex generic products. The focus on complex generics is critical because these drug products provide important therapies to patients and also are becoming increasingly significant to the economic health of the generic drug industry. For example, in an article published in February 2021, the expected drug savings from GX competition for 7 complex products, assuming generic price discounts from 30 to 44% and generic market share 40 to 60%, the expected savings from complex GX was approximately 25% of annual brand sales. Now I'd like to go briefly into some areas of opportunity for additional research considerations that could help advance the development of complex generics. Regarding synthetic peptides, referencing to rDNA RLD, FDA published final guidance in May 2021. However, FDA guidance did not provide scientific evidence in the guidance regarding what the numbers 0.5% and 0.10% were based upon, and this presents an opportunity for further guidance and research in this area. For example, can the regulatory impurity limits be based on solid scientific justifications that could potentially allow for higher level of limits without compromise to the safety and efficacy of specific peptide drug products? Further active research in this area is encouraged. And further opportunities exist for synthetic peptides regarding immunogenicity. For example, in future, can FDA guidance provide clear immunogenicity risk assessment pathways or flowcharts that industry can follow for in-silico, in vitro, or in vivo immunogenicity evaluations, and can further detail into the insights and outcomes of the completed grants and contracts in this topic area. And next, we'd like to briefly highlight opportunities for liposomal products. Although 2 liposomal generic products have been approved, doxorubicin liposome and amphotericin B liposome injection, some further opportunities include establishing guidance related to in vivo, in vitro correlations for liposomal products, mechanistic studies, investigations as to how parameters such as particle size distribution, in vitro release of a liposomal product affect its in vivo behavior. Moreover, establishing guidance on manufacture of aberrant formulations such as negative controls that could be used for testing the discriminative power of the IVR method. Establishing product-specific guidances with regards to PK modeling as a substitute to clinical studies for products where it is very difficult to recruit the patients would also be beneficial. And in regards to encapsulation efficiency, the amount of free versus encapsulated trap-bounded drug, a critical quality attribute of liposomal products, it would be very useful to have a guidance for development of analytical method or methods for determination of free drug in such drug products, and the parameters to be investigated and the validation expectations based on the research conducted. Now I would like to move to injectable and ophthalmic suspensions. More recently, there was an approval for a complex ophthalmic suspension, cyclosporin ophthalmic emulsion, 0.05%. However, there's still many complex GX products in this area not approved, and opportunities for research include understanding the controlling parameters of dispersion state, for example, paliperidone 1, 3, and 6 month, that govern not only drug absorption, but also stability, and development and establishing a suitable ex vivo permeability model to correlate with human in vivo PK profile for ophthalmic suspensions. And now moving on to nasal sprays. Since the publication of FDA's 1998 draft OINDP guidance and FDA's 2003 draft nasal spray guidance, the set of in vitro tests remains the same, also in the most recent product-specific guidance. Tests as spray pattern, droplet size distribution, and plume geometry are measured on fully developed spray in obstacle-free space and at distances which are far from conditions within the space-constrained nasal cavity. So an opportunity exists for the development of new in vitro tests with more relevance to anatomical, physiological restrictions of nasal cavity, such as nasal spray impactor. And now please let us discuss a few opportunities for drug-device combinations. For example, opportunities exist for connected devices for GX and alternative approaches for CEBE studies. Also, for CFD modeling, this was widely studied for liquid products and has demonstrated to be a useful tool for the characterization of MDI, nasal spray, and soft mist. However, little research work has been done with solid products such as DPI, and a further research opportunity is recommended. Next, we'd like to highlight some opportunities in model-informed drug development. MIDD is a topic that is well publicized by FDA through publications and meetings. It is widely used in formulation selection, BE trial simulations, bio-waiver requests, and sending specifications. Dr. Rebecca Jareb will speak more on this topic later in session 2A. There are clearly many opportunities we see for the FDA to pursue more research on MIDD. Dr. Raja Velagapudi will further expand on this topic in his public comment later in session 3. We feel aligning with other regulatory bodies on the expectations and utility of MIDD in regulatory submissions will be a good idea before research is initiated. In the next section, we would like to provide some commentary on exploring in vitro and modeling tools for respiratory products. Since respiratory products are very complex in nature, establishing IVC becomes a challenge for generic product development. Deposition and absorption from different lung regions is very important to achieve local and systemic drug concentration. As of now, establishing correlation with available in vitro tools like in vitro deposition, biopredictive, or discriminatory dissolution methodologies with respect to local and systemic concentration is not so predictive enough to understand the performance and equivalence of the generic products. Some roadmap on an alternative tool such as modeling approaches to replace some of the in vivo studies would be highly appreciated. It would be of great help to the industry if some guidances are established on IVIVC and use of modeling on respiratory products. Now, we'd like to move on to some opportunities for process modeling and automation tools. While there are many in silico and automation tools to better understand the process, we're still required to do the actual experiments, analytical testing as per the requirement. Opportunities exist to better understand the FDA expectation on use of software and validation of them for process modeling purposes of unit operations and stability modeling. And the research from FDA in demonstrating examples of such tools would be beneficial so that the potential of these tools can be leveraged upon without having to do extensive physical prototyping and analytical testing every time. And lastly, we'd like to highlight some opportunities in the AI and machine learning space. Exploring innovative tools such as artificial intelligence and machine learning for optimizing program design and reducing the study burden can be truly attractive for the GX industry. Could research in these futuristic approaches be performed, as this could help to establish the framework for including insights from such data-driven technologies into regulatory submissions? And next, let us discuss some opportunities as part of the GDUFA science and research journey. Could there be opportunities to share information with interim outcomes earlier in a more structured way, with enhanced visibility, and include more details about the research performed? Let's think about that. I would like to brainstorm with you some potential solutions for consideration. Notifications of when results are published in special research area and if they will be presented and where and when. Create and maintain a live database of outcome of FDA research as they get published. Relevant publications, summaries, posters. Could there be a dedicated event, virtual plus live symposium showcasing the outcome of GDUFA funded research and the ability to interact with the researchers? Could there be a database of key articles by product type, specific complex drug product, that could be reviewed and referenced by GX companies in preparation for control correspondences, product development, and pre-submission meetings with the FDA? And now we'd like to conclude with some closing remarks. Although some complex generics have come to market in the last few years, a significant number of important complex products are off patent, but still lack generic competition. With more complex products on the verge of losing exclusivity and patent protection, it is important to achieve demonstrable progress in increasing access to complex generics in the U.S. market. U.S. FDA is actively encouraging drug companies to take on the challenge of developing and launching complex generics, and there is existing opportunities for further research and to enhance dissemination and access to knowledge gained through the generic product science and research program, which we will further enable a more robust ecosystem for advancing complex generic product development. Thank you for your kind attention, and now I would like to acknowledge my colleagues from Sandoz who contributed in the preparation of this presentation. Thank you, and wishing you a great rest of the workshop, and looking forward to our panel discussion on day 2.